It says we use the term homophytic and heterophytic for every terrestrial plant, from mosses to flowering plants. But there were a limited number of plants for which we use the term monoecious and dioecious. You might remember in the lecture, I didn't really talk about the words monoecious and dioecious until later on, right? I talked about homophilic and heterophytic, and then it was probably a week later before I came up with the terms monoecious and dioecious. So, of course, I'm not just going to ask you to define the words, I'm going to ask you why, because I want you to show me that you really understand. So, but let's start with what the terms mean. So what does homophilic and heterophytic, what do those words refer to? They refer to the gonidophyte, right? And they refer to whether or not the Antheridia and the Archegonia, the two gametangia that produce sperm and eggs, are found on the same individual or on separate individuals. Now, why would I have to start with those terms right at the very beginning? Because every plant makes two different gametangial types, right? We didn't talk about some kind of a plant species that makes only sperm and never, ever, ever makes eggs. There's no individuals that make eggs, they make only sperm. Because sperm don't fuse with other sperm, you realize, right? Eggs don't fuse with other eggs. So every species of plant that we talked about has to have two different types of gametes, two different types of gametangia, right? One that's going to make sperm, and then one that's going to make eggs. And then we can talk about whether the sperm-making gametangium and the egg-making gametangia are on the same individuals, in which case it's homophilic, or whether they're on separate individuals, in which case they're heterophylic. But every plant species makes sperm and eggs, so you kind of need those terms right off the bat, right? How about the words monoecious and dioecious? What do those mean?
On the same plant or a different sporophyte?
Yeah, those are sporophyte-specific words, and what they refer to is whether or not microsporangia and megasporangia occur on the same sporophyte or on different sporophyte individuals, right? Now notice that's only going to be relevant. Those terms are only relevant if you first have a plant that is heterosporous, that has both microsporangia and megasporangia. Otherwise, you can't use those words. It makes no sense to talk about whether or not a plant has microsporangia and megasporangia on the same individual or on separate individuals when there are no microsporangia and megasporangia. There's only one sporangial type. So for a plant that is homosporous, you would never use the words monoecious and dioecious. And the reason I didn't present those words to you in the beginning is because when we were talking about mosses and liverworts and almost all of the ferns and almost all of the lycophytes, they're all homosporous, right? With the exception of Selaginella, Isoetes, Marsilia, Salvinia, and Azolla. With the exception of those five, they're all homosporous. So the words monoecious and dioecious don't really even come into play. Could you all write that out? Basically, the short answer is every plant species, whether it's no matter what kind of a plant it is, has two different types of gametes, has sperm and eggs. So it's relevant to talk about whether or not those two different gamete types are produced on the same individual or on separate individuals. But only some plants have two different spore types, right? So it's only when you start talking about heterosporous plants in detail that the words monoecious and dioecious become relevant. They're not used otherwise. Yeah? Why can we apply a label like homophilic or heterophylic to every plant species, and we can only apply the labels monoecious and dioecious to some plants? Because only some plants are heterosporous, but every plant has two different gamete types. Okay, so on one of the free response things for the, I'm going to give you one of the answers to the actual exam I'm going to give you. On one of the, didn't it say that? It said at the beginning of Lab 6, come on, somebody that took the practical, we all took the practical, somebody that, I can't remember whether it was Wednesdays or Tuesdays, but on one of them, the free response thing said, at the beginning of Lab 6, I make a big deal about life histories. Why are our life histories so important for plants and not as important when you're talking about fungi and animals? Did you all read that question? Yeah, that was, okay, that was yesterday. That was the Wednesday one. Okay, so you know what the answer is to that. Why is a life history so important? Why is it that when we're talking about plants, it just becomes completely impossible to talk about them at all unless you have a clear understanding of a plant life history? I could talk quite a bit about fungi and about animals, and if you don't really know the life history, it's not that big a deal. But why, when it comes to plants, is it such a big deal? Anybody try to answer that one? Anyone want to guess the answer to that?
You have an alternation of generation.
Because they have an alternation. That's, you're on the right track.
2 pages on that one.
You're on the right track. When you say there's a haploid phase and a diploid phase, but animals and fungi have haploid and diploid phases too. What really makes a plant different?
They're both
Right, because you have haploid cells too. They're gametes, right? And fungi have haploid cells, or have diploid cells, the zygote, right? So you have a haploid and a diploid phase too. But why is it not so critically important in talking about you? Like I can talk a lot about your biology without running into trouble, but when it comes to plants, it becomes a big deal if you don't understand the life history. It's because,
Alternation generation.
yeah, which means what? What does alternation generations mean? It means not just that there's a haploid part and a diploid part, but that, what can you say about each part?
They're adults, they're
They're
mature.
adults. There are two different adult forms, right? You have only one adult form. All animals have only one adult form. So if I start talking about an animal, right, like I start talking about a tiger or something, you're assuming I'm talking about that adult, right? Because there's only one of them. But when you're talking about plants, you have two multicellular adults. It becomes very important to know which part of the life history you're talking about because there's two radically different adult forms. One adult is diploid and doing meiosis to make spores. The other one's haploid and doing mitosis to make gametes. Those are two radically different adult forms. When I'm talking about fungi, there's only one haploid adult individual, so it becomes easier to talk about fungi because no matter what I say about its biology, you know that I'm talking about that one adult. If I'm talking about an animal, you know I'm talking about that one adult form, right? But in plants, there's two separate, very different adult forms. And so, like, for example, these words, like homophthalic and heterophthalic, they refer to only one of those adult forms. You can't use those words if you're talking about the sporophyte. And the words monoecious and dioecious refer to the other adult form. So you have to keep track. That's why in Lab 6, I spelled out the terms and said these words are words that refer to the gametophyte. These words are words that refer to the sporophyte. If you're not even aware that there is this alternation of generations, that there's two multicellular adult forms, it's very hard to talk about plants. You really have to keep track of, are you talking about the gametophyte or are you talking about the sporophyte? In the case of fungi and animals, it doesn't really matter because there's only one adult, right? So if I talk about a fungus or I talk about an animal, I'm talking about the adult, right? I'm probably not talking about the gametes or the zygote, right? I'm talking about some aspect of that particular adult individual, or maybe talking about it as a juvenile becoming an adult or something, but it's clear what I'm talking about. There's only one generation. There aren't two different multicellular generations. Yeah, okay. So you realize you're going to have something exactly like this. It's just that I'm going to take, this is one of the animals you're going to see next week. This is Obelia. It's a colonial hydrozoan. I'm going to delete Obelia, and this is Fucus down here. I'm going to delete Fucus, and I'm going to put two other life histories in here that you've never seen. But you're all going to get perfect 20s on this, right? Because you understand the trick. What I would do is I would come in here, and on yours, I'm going to make it straightforward so that it will say meiosis, and it will say fertilization, or syngamy, or something like that, that leads you to think sex, right? Hopefully, it leads you to think sex. I would circle those. That's the way I'd start this. I would circle where it says meiosis, and circle where it says fertilization or syngamy. That's the first step I would do. If it says mitosis, like you'll notice this one says mitosis all over the place, I'd completely ignore that. Because that has nothing to do with determining whether or not you're diploid or haploid. Notice the only thing it asks you to do over here is just identify each phase as being diploid or haploid. This is ridiculously easy. If you are after meiosis and before fertilization or syngamy or karyogamy or whatever you're calling the sex, you are haploid. If you're between syngamy or fertilization or karyogamy and meiosis, you are diploid. Now, unfortunately, I've had some people learn this exactly backwards, so they get every single one wrong. Try not to do that, right? Try to remember if you're coming out of meiosis, you're haploid, right? So if you're after meiosis, that's the haploid one. So now that makes this trivial and easy. Even if you've never seen this life history and you know nothing about Obelia at all, hopefully when you look at this, you might kind of realize this is an animal. It has an animal life history. The only haploid part of an animal life history are the gametes. Not to mention the fact that anytime I label something that's an egg or a sperm, as far as you guys are concerned, that's always going to be haploid, right? I'm not going to show you a diploid gamete of some kind. Okay, so let's go around. So number one is on this little swimming planula larval stage that it goes through, right? But you know that that's after fertilization, and the cycle goes this way, right? It even has little arrows to show you which way it goes. You're after fertilization, and you're way before meiosis, right? So it obviously must be diploid. So all you do is write on line number one there, you just write diploid or 2N, right? How about now, if I plug along here and over here we have a feeding polyp, that's obviously going to be diploid. A reproductive polyp, that's obviously going to be diploid. We have the medusa up here, which is the adult form. Notice that the polyp is a juvenile form. The adult medusa is going to be diploid, and then I have 5 on the egg. That's obviously going to be haploid. And even if for some reason you couldn't remember that eggs are gametes and that they're haploid, it's right after meiosis and it's right before fertilization. And notice that the sperm and the egg are the only thing in that whole diagram that I could label it would be haploid, right? So on these lines, all you have to do is write diploid, 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 haploid. There you go, you got 10 points. You see you're all going to get 20 on this, right? You shouldn't get anything but 20. This is supposed to be the easiest one. Okay, now we come down here to fucus. You might look at that and go, wow, I don't know anything about fucus at all, but that's okay. You see where the meioses are? There's a meiosis right here, and there's a meiosis right there. It's showing you because it separated meiosis in the female and in the male, right? But I would circle those, and then I would circle fertilization, which is up here. So now to ask you 6. 6 is on the sperm. Again, you might sort of guess that sperm are haploid. So on line 6 you should write haploid. But even if you didn't know that, you would see clearly that 6 is between meiosis and fertilization, right? It's after meiosis and before fertilization, so it has to be haploid. How about 7? 7 is coming from the zygote. It's after fertilization, so that's diploid, right? 8 grows up into the adult, right, which is again after fertilization and before meiosis, so that would also have to be diploid. 9 is on the conceptacle. It's showing it as being pre-meiosis, so these are including the cells that are going to undergo meiosis in order to make eggs, potentially, or sperm. So those would be diploid. And even here, the way 10 is labeling the antheridium in this case, presumably, I actually don't like this. To me, fucus has an alternation of generations. It just has highly reduced gametophytes that are contained inside the conceptacle. But you don't have to worry about that. Instead, you look at this and you just go that the antheridia are what? Prior to meiosis. It's showing meiosis occurring inside of the antheridia to make sperm, right? So it would be diploid.
But those spores that come down, that grow back up, the new gametophytes are going to be...
Yeah. So in the case of fucus, a lot of people, like for example, if you were taking this course at San Francisco State University, they would show you this and tell you this was an animal life history. Because it kind of looks that way, doesn't it? It kind of looks like you have these antheridia that are diploid, they do meiosis in order to make the sperm, which are haploid. Isn't that what that looks like? That looks like an animal life history.
Yeah.
The problem of this, so here's where, now I'm going to give you the added little piece. I wouldn't ask you this, but I'm going to give you the added little piece. The cells that come out of the meiotic division inside of those antheridia divide mitotically. Does that bother you?
Well, they're going to make spores, they're going to grow from the new gametophytes, right?
Well, okay, so my interpretation would be those cells coming out of that meiotic division are not sperm. Sperm cannot divide, right? So my interpretation would be that those are spores.
Yes.
The spores are contained inside of that conceptacle and develop inside of the conceptacle and undergo several rounds of mitosis and then start producing the sperm, which are liberated from the conceptacle. So my interpretation of that would be that this approach is something kind of like what seed plants do, where the spores develop inside of these sporangia and then produce gametes, and the gametes are released into the water. So I would interpret this as an alternation of generations. If you ask me, I would tell you that all brown algae, all Phaeophyta, have a plant life history. But there are people that interpret this as an animal life history. I think that's wrong, because if you do that, you have to accept the idea that gametes can divide, and I don't accept that idea. So
Don't they have, whenever the antheridia makes this, once they make the spores and whatnot, don't they have to grow up into new gametophytes before they can make gametes?
right. I would not call these antheridia. I would call them microsporangia.
Yeah. So it's two different types of spores.
I'm showing you a life history I don't really accept as right.
So that's going to...
But it's in, you realize, like, this stuff doesn't come down on a mountain with Moses or something, right? This is all a matter of interpretation. It's a matter of how different biologists look at this and interpret this. I would not interpret this way, right? I would not call these little structures inside of the... Well, I mean, in all honesty, the way that they've shown it here, I think actually the meiosis has occurred before these structures that they call antheridia. Either that, or if these little structures really are diploid and they're doing meiosis right here in order to make haploid cells, they're not making sperm. I know for sure, because I've looked this up a bunch of times because this bothered me, the products of meiosis in fucus undergo mitotic division. So my interpretation of that would be that they're spores and that they do a few rounds of mitosis and then produce gametes from that. That's all way more than you guys need for this, right? All you need to do is you would come to me and you'd say, hey, 10 comes before meiosis, it's diploid, right? And I'd take that. That's the answer I want you to write. All I want you to do is figure out where meiosis and fertilization are and figure out whether it's haploid or diploid from that. You don't have to sit there and try to critique the, you know, the subtleties of the life history and whether or not it's drawn correctly.
Is this homosperous homophallic?
Well, actually, you have both the male and female in the same conceptacle, so.
So it is homophallic.
Yeah, right. it would
But
be
is it
homophilic.
homospecious? Because I can't tell from anything.
Well, the problem is, see, I wouldn't even use the word homophilic for this, because this individual's diploid, so I would consider this to be the sporophyte. But if you're gonna take this as being an animal life history, then you would say that it's bisexual or homophilic, if you were gonna use that word for it, yeah. Yeah. Okay, not to bog you down, is everybody convinced they could get 20 on this? Yeah. Because you're just gonna circle where's meiosis, where's fertilization. If you're between meiosis and fertilization, you are haploid, and if you're between fertilization and meiosis, you are diploid. Maybe write that in your cheat sheet. Okay, how about 8? You all recognize that diagram? So it shows you the life history of ova. C letters, right? Let's just look at it real fast. You know where I got this from, right? I got it right out of the slides. Here it is. So it says, ova lives in the ocean, right? It's an alga, it's not terrestrial. What aspects of the ova life history are similar to the life history shared by all terrestrial plants? I've noticed before, so there's sort of 3 different groups we're talking about. Terrestrial plants. This would be like land plants, is the other way to say this. Embryophytes. If you said plant kingdom, historically, this is what we're talking about. Mosses, liverworts, hornworts, ferns, lycophytes, and all the seed plants, right? Then we have this other group, seed plants, which have some non-flowering seed plants, gymnosperms, right? Things like cycads, natales, ginkgo, conifers, right? And then we have flowering plants, which would be just the angiosperms that make flowers and fruits. People confuse these. This group is inside of this group, right? This group is inside of this group. This is the most inclusive group, yeah? This doesn't belong to any of those groups, right? This is outside of these guys. It's an alga, it's not a terrestrial plant at all, yeah? So let's try this. So the question is, what aspects of the ova life history are similar to the life history shared by all of these guys?
Isn't it the sister group to that monophyletic group, though?
Yeah, this probably isn't, remember. It's probably the charophyte algae, not things like ova.
It's still a green algae, though. But it's,
But it's
only
still
the
a green algae.
relative.
So it's not the sister group to this, but it's in the Vertiplantae. It's somewhat related to it. So I know some people in the other class, you know what they wanted to put there? They wanted to put things like, oh, it has chlorophyll B, right? Which, that's true, it has chlorophyll B, but it says what aspects of the life history. So let's focus, that's not part of its life history, right? That's secondary photosynthetic pigment, right? So instead, let's focus on its life history. So what about this life history makes it look plant-like? It's not a plant, but what makes it look plant-like?
Alteration.
Okay, it's an alternation of generations. That's the thing that should most jump out at you. There's a multicellular haploid phase and a multicellular diploid phase, right? The haploid phase makes gametes, the diploid phase makes spores by meiosis. That's probably it. Is there anything else you want to put there? So now the question is, what aspects of this life history are different from plants? Now there's one you should definitely get and another one that you might not pick up on, but I'll point it out to you.
Gametes and release.
Both gametes are released. And that's never going to be the case in any terrestrial plants. If this gamete happens to be the egg, remember that the egg would be retained inside of the female gametangent, inside of the archegonium, right? It's never going to be released. The sperm are released, but not the egg. So that's one of the very key differences, right? The egg is not being retained inside of the female gametangent the way that it would be in every single one of the seed plants, right? Anybody pick up on another difference there? Like if you look at this,
Gametes have flagellae.
the gametes have 2 flagellae here, but a lot of the gametes you realize are flagellated in terrestrial plants too. So for example, they have 2 flagellae in Bicon too. So for example, things like moss, sperm, swim in water. So do ferns, sperm, swim, right? Cycads, some of the cycads, believe it or not, have flagellae, like thousands of flagellae on a single sperm. They swim through the little pollen tube, if you can believe that. So, yeah, they tend to be flatulated as well, at least the sperm, not necessarily the eggs. The eggs are, I don't think there are any eggs that are flatulated in terrestrial plants, but the sperm would be. But something more about the overall life history, like maybe here,
The same.
they look the same. This is called an isomorphic alternation of generations. You can't tell which one is the gametophyte and the sporophyte just by looking at them, right? You'd have to sit there and count chromosomes or something in order to try to tell them apart. What do you think we call the type of alternation of life, of generations that you have in terrestrial plants? Iso means same, right? Like an isosceles triangle has two sides that are the same. Same, and morph means form, same form. The gametophyte and the sporophyte look exactly the same. What do you think you call it in terrestrial plants? Heteromorphic, right? Like think in every case, right? Try to picture what do the gametophyte and the sporophyte look like in a moss. Can you picture them? The gametophyte's that little green stuff, right? And the sporophyte grows out like this and has a little capsule. And they don't look anything like, right? If you think about what the gametophyte and the sporophyte look like in a liverwort or in a fern, it never looks the same, right? They're always different. You could easily, if I showed you a sporophyte or a gametophyte in any terrestrial plant, you can identify it as being the sporophyte or the gametophyte. That's not true here. Here, if I showed you ulva and I asked you, is that the gametophyte or the sporophyte, there'd be no way to tell.
So is all green alga isomorphic alternation generation? Or
No,
is it just
no,
ulva?
no, they're heteromorphic ones too.
So this is just ulva that has this?
Well, not just ulva, but there are other green algae that would too, but in this particular case, it has an isomorph. I'm trying to think of another one that does. Most of the red algae and brown algae that have alternation of generations are heteromorphic. Green algae, I'd have to think about it a little bit more. But there are at least some that are isomorphic. Yeah. I don't know that you pick up on that one, but the idea that the female gamete's not retained on the female gametangio, that you should definitely pick up. So, could we get those? You know what people will write here is it'll say, what aspects of the oval life history are different from the life history shared by terrestrial plants? And you know what I've had people write in there? Because you realize I used this exam before. You know what they'll write? They'll say, ulva uses water to transport sperm and terrestrial plants don't. Is that true?
Birds use water, no.
Yeah, ferns use water. So do mosses. So do liverworts. Those are all terrestrial plants. What are they thinking? They're thinking seed plants, right? Sometimes I'll ask them, seed plants are awesome. Why are seed plants awesome? And you know what they'll write? Because they don't make food inside of the seed until a baby comes. Is that true? That's not true. Not all seed plants, right? They're confusing these guys, right? So if I ask you, why are angiosperms so awesome? Now maybe you'd say, oh, because they use endosperm as the food reserve, so they don't make a baby inside of their seed until after they're sure that they don't make a food reserve inside of their seed until after they're sure they got a baby, right? And also they have flowers that can attract all different kinds of pollinators, and they have fruits for all different kinds of cool dispersal of the seeds and stuff like that. How about why are seed plants so cool? They don't need water for reproduction, right? Because the whole male gametophyte travels and builds a little pollen tube and passes sperm through the tube, right? And also because they have this resistant structure called a seed, so that the little baby sporophyte doesn't have to grow and develop immediately, right? It can actually stay dormant inside of the seed. If I asked you why are terrestrial plants so awesome, what would you say? Don't say something about that they don't need water for reproduction, because that's not true. A lot of them do, right? But maybe here you'd be talking about the fact they have cuticle, they have stomata. Those are specific adaptations to being terrestrial, right? You would never find cuticle or stomata in algae. So you have to keep track of which group here you're talking about, right? Because otherwise you're going to give weird answers. I know some of you are going to write that terrestrial plants don't need water to reproduce. I know you guys will write that. Yeah, seed plants don't, but ferns and mosses, those are terrestrial plants, right? And they very much do need water to reproduce, so that's not right if you write it that way. Okay, so here, number 9 says all terrestrial plants, meaning all terrestrial plants, right, have relatively small gametophytes. The tallest terrestrial plant gametophytes are only a couple of inches tall, right? So if you think of something like a moss or a liverwort, it's about this big, right? There's no moss or liverwort that's like 6 feet tall. You realize that, right? And think of the ferns. Some ferns get very big, but not the gametophyte, right? It's the sporophyte that's getting really big. And if you think about like all of the bushes and trees and big plants that are outside, those are all the sporophytes. What do the gametophytes for all of those plants you can see outside look like? They're really, really tiny, right? In the case of most of them, they're seed plants, it's a pollen grain or something. It's like 2 little cells. And the female is tucked away inside of the megasporangium, inside of the seed, right? Or what will be the seed in the ovule. So all of those guys, the gametophytes, are really teeny tiny. And in the case of even things that have the gametophyte as the dominant part of the life history, like a moss or a liverwort, mosses, I mean, maybe the biggest mosses get kind of like this, right? They don't grow into some big bush or anything. So the question is, why is that?
But those gametophytes are still much larger than the ones that are the seeds, right?
They're much larger than the seed plant gametophytes, but they're still relatively small by gametophyte standards. Like, look at these gametophytes. Those are big, right? These could get to be about like this.
Holy cow.
They've got a big gametophyte. So the question is, why is that? Why is it that terrestrial plants, why is it that the gamete-producing individual has to be very small? And why is it that the gamete-producing part of an algal life history, like think of the charophyte algae, remember they have like a fungal style life history? So the haploid part of their life history is huge. That's the big, large, long-lived part of their life history. Why do algae get away with having the gamete-producing part of their life history being really big, and terrestrial plants are forced to have it be very small? Think about it for a second. Transportation of sperm, and sperm don't fly, so they have to have water. So if I'm an alga, where do I live? In water, right? So I'm an alga. I'm 60 feet tall, but I live in water. I could be 60 feet tall. I could just make sperm up here. Let them go. Go into the water, right? If I'm on land, and I'm like 6 feet tall, and I'm trying to let sperm go into the air, that's not going to work very well. So how come I have to be really small? Because if I'm really small, what's going to happen every once in a while? I'm going to get covered over with water. Right? If it rains really hard, you know how you can have that much water on the ground, right? When you get that much water over the top of a moss or a little fern gametophyte or a liverwort or something, it goes, woohoo, I can have sex, right? It starts letting sperm go, right? But if you're 6 feet tall, if you had a moss with a moss antheridial head that was up here 6 feet tall, it'd have to wait for like Noah's flood or something. How often are you going to get covered over with water if you're 6 feet tall? Not very often. But if you're only this big, I mean, all you need is a good hard rain and you're going to get covered over with water, right? And in the case of seed plants, the gametophytes have to stay very small. Why? They have to be even smaller, because now not only do they have, it's not like they just have to be small enough to get covered over with water, they have to actually be small enough to do what? To let the wind carry them somewhere, or to be able to grab onto a butterfly or a bee or a hummingbird or somebody that's going to carry them. So they really have to be small. So that's the answer here. You've got to be small because you've got to be small enough to get covered over with water. That's not a big deal if you live in the ocean or you live in a lake or something like that. But we're talking about terrestrial plants. You live on land, and yet you have to be covered over with water. That's not going to happen unless you are very small. If you get huge, you're not going to get covered over with water. Not regularly, anyway. Yeah?
That was probably an evolutionary adaptation as well, do you think? Because they probably,
It would
back in
have
the
to
day,
be.
they probably could
So-
have been really big, and then over time they got smaller.
Not even back in the day. What's the sister group to terrestrial plants?
The green alga,
Which green algae?
the closest relatives.
Which are charophyte algae. And the charophyte algae have a fungal life history. When you look at a charophyte alga, everything is haploid. Everything is the gamete-producing part of the life history. The only diploid part of the life history is just the zygote. So if you think about it, we went from something that's all haploid all the time, with the exception of the zygote, this huge haploid thing living in water, making gametes, right, and just letting them go, except for the egg. Remember, it hangs on to the egg. This is part of the reason hanging on to the egg is important, because when you go into a terrestrial environment, at least you don't have to worry about the egg swimming anywhere, right? Because the egg is going to stay there. So at least you're retaining one of the gametes. If you were something like ova and you're trying to move on to land, boy, you got a problem. Your eggs are trying to swim, your sperm are trying to swim, you're in big trouble, right? At least these guys, when they go on, the egg doesn't have to go anywhere, but you still got to release sperm into the environment. So somehow we had to go from being big haploid to being little haploid. And then at some point, in order to completely free the life history from water, we have to get really little haploid. So think about it, the most basal plants have an alternation of generations, but the haploid part of the life history is the dominant long-lived part of the life history. It has to stay small though. It had to get significantly reduced. So we go from the charophyte algae that are haploid all the time to something that has an alternation of generations. Presumably you need to do this in order to move in a terrestrial environment, but get the haploid gamete-producing part of the life history small. By the time we go to ferns, we now have the sporophyte, the diploid part of the life history is now the long-lived dominant part. The gametophyte gets smaller still, and by the time we move into the seed plants, the gametophytes have been reduced really tiny so that you've completely freed the life history from water. This is why I said you had to understand plant life histories in order to understand major trends in plant evolution. If you look at how green algae, in effect, moved into terrestrial systems and then dominated them and completely terrestrialized themselves by freeing their life history from a need for water, it has to do with making the haploid part of your life history smaller and smaller and smaller so that you're not dependent on releasing gametes into the environment, right? In fact, if they went any smaller, right, they'd have an animal life history. They've gone from something that was completely haploid to almost something that's completely diploid. Think about the flowering plants. The male gametophyte's only 2 cells. If you didn't do that one mitotic division, you'd argue that this was a sperm. You'd be making sperm by meiosis, right? It wouldn't be a spore that does a mitotic division to make 2 cells, one of which does a mitotic division to make 2 sperm, right? Instead, it would just be a sperm. You're doing meiosis to make sperm. It's the only haploid part of the life history. It would be an animal life history. They've pushed it as far as you could possibly go. They've gone from something that basically is a fungal life history all the way to the side of something that's like an animal life history, through all of these different transitional forms where the haploid part just keeps getting smaller and smaller and smaller and smaller. Why did they do that? The reason they did that is because if you're on land, you cannot have this huge haploid thing that you're releasing all these gametes that have to swim into the... Notice fungi, sure, they have a haploid-only life history. They don't have flagellated cells anymore. They don't release things that have to swim. So different strategies as to how you move into terrestrial environments, right? But this is the way plants did it. Yeah, because
So all of these plants had to have originated from being in water, you know what I mean? Like all of the land, like all the big pine trees and all the trees out there.
Well-
you think they come from an ancestor that was definitely in water? I mean, it makes sense.
And humans.
all reconstructions, all phylogenetic reconstructions put terrestrial groups back with aquatic origins. So life forms typically started in aquatic, and interestingly enough, the closest relatives of terrestrial plants, they're not ocean-dwelling things like ulva, they're freshwater.
Freshwater green algae relatives?
The charophyte algae are freshwater.
Oh, wow.
Interestingly, terrestrial vertebrates, like reptiles, birds, and mammals, their closest relatives are also actually freshwater. So it's not thought that terrestrial invasions happen from marine systems, but from freshwater systems. There's quite a bit of evidence, for whatever reason. And obviously happened differently in different lineages, but we're looking at something analogous there, right? But in fact, towards the end, I'm going to ask you what group in terrestrial vertebrates is analogous to non-seed plants, and the answer is amphibians. Things like frogs and salamanders are terrestrial, but where do their eggs go? In the water, right? They're dependent on water for reproduction. Even when you see the frogs, you know that amphiplexus or whatever it's called when the daddy frog gets on the mommy frog and it looks like they're copulating, you realize they're not, right? He's actually just squeezing the eggs out of her into the water and then putting his milk in the water behind her so they have external fertilization. Basically, frog eggs are kind of like fish eggs. They're basically fish eggs. What group is analogous to seed plants within terrestrial vertebrates? The amniotes. Reptiles, birds, and mammals. If you look at a reptile egg, which is basically a chicken egg, it's a land egg. Even reptiles that spend their entire lives in the ocean, like sea snakes and sea turtles, where do they lay their eggs? On land. And that amniotic egg is analogous to a seed in the sense that it allowed terrestrial vertebrates to become fully terrestrialized and to free their life history from a need for water. Amphibians haven't done that yet. Amphibians are terrestrial, but they're still tied to water for reproduction. Okay, I digress. So did you get this? The gametophytes have to be small because they're releasing sperm into the environment. That means you have to have water. If you're this big, you're much more likely to get covered over by water in a terrestrial environment than if you're this big, right? Algae don't worry about being little so that they get covered over by water because they're covered over by water all the time. No matter how big they are, they live in water. Yeah. So if you live in water, you've got very few problems with respect to this, right? There's no problem with worrying about drying out, or worrying about gas exchange, or worrying about transport. You realize when I was talking about xylem and phloem, like specialized tissues that move food and water around inside of a plant, you realize algae don't have stuff like that, right? They don't need stuff like that. Because you're bathed in water all the time. You don't have to worry about bringing water in just through your roots and moving it up through your stem to all of your leaves and stuff. Why do you have to worry about that? You're just bathed in water. You just take water in all over your whole body, right? Life is easy if you're an alga. You also don't have to worry about being rigid and being able to stand up. As long as you're buoyant, you just kind of float, right? So all of these really important lignified tissues and everything that you find in woody plants that allow them to stand up, you don't find any of that in algae, right? There are some semi-woody tissues, but nothing that's truly woody because they don't need to stand up in a air environment. They just float and they're buoyant, right?
Now, how does the gametes find the egg in the case of like ulma and
Weren't you the one that asked me that before or no?
Well,
Somebody,
yeah, but it
one
was-
of you guys asked me and I ended up
yeah,
looking
I did,
at
but it was in like, I think it was.
Didn't I say it was chemotactic or something I looked up?
But
Basically,
but that was for like, I think it was a non-seed plants, I think that was.
yeah, but it would be something similar to that.
In the
There's some
water.
kind of, right, there's some kind of a chemical signal that they use that they can recognize
I was
each
just
other
gonna
in
see
there.
if there's any difference there, I mean,
Yeah, no, I mean, yeah, I'm slightly lying, look it up. I don't know for sure, I'd have to look it up in order to be sure, but I would assume that there's some kind of a chemical sensory system that they use. They're probably secreting something, and I don't know whether it would be pheromones exactly, but something that's some kind of a chemical cue that draws the gametes together. Okay, are you all satisfied with the answers to these? Now, do you have anything else? Are you all kind of getting this, kind of pulling it together? You realize that this part doesn't go back to the very beginning, right? It only just goes stuff we did since the last exam. So origin of life stuff, prokaryotes, protists, fungi, and plants. And you all get a cheat sheet, right? So you're making a cheat sheet.
What are some of the highlights on fungi? I know it's a broad question.
Okay, so
What-
like on here, let's see, the one question on fungi here had to do with the spores, right? Okay, I'll give you insight. You want to know how I make the exams? I haven't written this one yet, so I'm still working on it, but you know how I do it? So I think, okay, well, there should be a question on fungi. And so what I do is I'd actually go right to the PowerPoint slides. This might be the way you should study, really, in the sense that you could open this up and think, okay, now what kind of crazy questions could this guy ask? Right, so I sort of told you what fungi are, all the different traits that they have. I could ask you something about that. Remember chitin cell walls? You don't find those outside of fungi. I could say something like saprolegnia, which you all know, right, is not a fungus. But it was historically treated as a fungus in some systems, right? Why? What characteristics about saprolegnia made them think that it was a fungus? And you could say what? What would you say? Like on that list, what things does saprolegnia have? Let's say that again. Okay, it's eukaryotic and heterotrophic, that's true. Has those, I didn't even think about that. How about that down here?
Absorbs nutrient.
It has filamentous growth and it has absorptive nutrition, right? In fact, I have asked this question before, or I've asked some version of this question, right? So, and then I would say, so how could it share those characteristics with fungi if it's not actually related to fungi? Because those characteristics probably represent, in the case of being eukaryotic and heterotrophic, those characteristics are ancestral, right? And these characteristics of being filamentous and having absorptive nutrition are probably convergent. Yeah. Then I could say, what's a characteristic that fungi have that saprolegnia doesn't have? Something that really sort of lets you know that it's not a fungus. So chitin cell walls would be a good one. Maybe if you said something about this lysine synthesis pathway being different. Yeah. You might even say something about the life history, right? It has more of an animal-style life history. Okay, so what else could I ask you? You know, a lot of times, you realize generally if I use a picture on here, it's with the exception of these life histories that I'm going to give you, but like this picture came right out of the PowerPoint slides, and typically I'll use it. So it's easy to just go like copy, you know, and then paste. And then I could say something like in the lab manual, I used a capital letter to refer to the glomerulomycota, the ascomycota, and the basidiomycota. Why did I use a lowercase letter to refer to chytrids and zygomycetes? Yeah, that's something you couldn't copy over, it's showing whether or not you understand it. So why did I use a little c and a little z when I used the word chytrids and zygomycetes based on this tree?
They're not a real.
Because they're not monophyletic groups, right? Chytrids and zygomycetes represent a bunch of different basal lineages and are represented in this particular tree as just a polygamy at the base of fungi. Okay, what else could I ask you? Sort of that question that's on here goes back to this, right? It's sort of asking you what's the difference between like a zygospore and an ascospore and a basidiospore, and throws in conidia as well, right? Like question, whatever one that is. 4, most fungal reproduction, dispersal, both sexual and asexual, is carried out via structures referred to as spores. What do all spores have in common? And then describe the differences between zygospores, conidia, ascospores, and basidia. It's kind of outlined there. What else could I ask? I know I've asked before, I've said something like, name one important symbiotic relationship between a fungus and an autotroph. So you could say, what's the name of the, I could actually just go copy paste. What's the name of this association? This I asked you on the lab. Endomycorrhizae or mycorrhizae. So mycorrhizae is, and then I said, and tell me something about the two partners. Well, one partner is a fungus, the other partner is the plant, right? And then maybe what does each member get out of the relationship? So a plant would be getting what out of this?
Water and minerals.
Water and
Phosphorus.
minerals, phosphorus especially. Plants are horrible at taking up phosphorus, but fungi do it very well. And then what does the fungus get out of it?
Sugar.
It gets sugar primarily. The other possible answer you could put would be this. That's a relationship between what? An alga and typically an ascomycete. It's a lichen, right? But that would be another symbiotic relationship between a fungus and an autotroph. I could say something like, many fungi are parasitic on either plants or animals. Name one fungal parasite. Tell me about it. What could you tell me about? Maybe you tell me about this one.
That's a hard one. Don't put anything like
Yeah,
that.
ergot, right? Clearly there, what, the fungus is parasitic on rye, right? And it produces this stroma that replaces the rye grain, and it can cause ergot poisoning if people ingest it. That would work. I've asked before why slime molds were considered fungi, kind of the way that I asked you why saprolegnia would have historically been considered a fungus. Why would slime molds have been considered fungi? Why would people have thought, you know what slime molds are, right? Like, people tend to mess this up too when I ask them. Why would people have thought these guys were fungi historically? It's not really because they have filamentous growth, because they don't, right? They go through an amoeboid stage, right? And they don't have absorptive nutrition. They tend to feed by phagocytosis. But what do they make here? They make little fruiting bodies that produce spores. That looks very fungal. It has nothing to do with fungi, right? But that's why. So I've asked before, why were slime molds historically considered to be fungi? Why did some people think that they were fungi? It's because they make little fruiting structures that look kind of like the fruiting structures that fungi make that produce spores. Yeah. I don't know, can you think of other questions? This is exactly what I'm going to be doing in the next day here, is trying to think of questions for you guys. But typically what I do is I come back to the PowerPoint slides. Yeah, you know what these are? What are those? Who is this?
Rhizopus?
No, this would be Rhizopus.
Oh.
This is Aspergillus, right? These are conidia. It's asexual, so I could tell you something like Aspergillus is an ascomycete, but we would never know that it's an ascomycete based on the conidia it produces. Why? Something like that. And why is that? Because fungi are assigned to those different groups, right? Based on what? Based on the Sexual structure, no, the sexual structures that they produce. Typically, ascomycetes make conidia if they're gonna make asexual structures, but you could never identify something as being an ascomycete based on conidia. You can't identify a zygomycete based on sporangia. You have to identify them based on the sexual structures. So if you have fungi that reproduce only asexually, I'd have to word that question better. I could say there historically used to be a group of fungi called Duromycetes that included individuals that only reproduce asexually. Why were they all thrown together as Duromycetes when we now know that they really belong with ascomycetes? And the reason is because if you only reproduce asexually, you can't be put into a fungal group based on asexual structures, right? Only based on sexual structures. We know that these are ascomycetes now because we have DNA sequence for them. And if you pop into a phylogenetic analysis, they pop out right in the middle of ascomycetes, right? They clearly are ascomycetes that have just abandoned sexual reproduction for whatever reason. I'd have to think about how I could word that so that you could actually get it.
The ascomycetes make sexual and asexual?
Some do, but these guys typically don't. Penicillium and Aspergillus and a lot of the molds will never ever make asci. I'm pretty sure there's a large number of them for which nobody's ever observed asci. Some of them you can kind of trick them into having sex and they'll make asci for you. And they will make asci when they reproduce sexually.
Well, all that is asexual, right?
Are typically all asexual too. Yeah, asexual reproduction in fungi is big, right? It's big. It's typically more common. And zygomycetes, this is an asexual structure, right? And rhizopus, like if you looked at those plates, you realize that Allomyces and stuff, you realize every fungal plate that you had, all rhizopus now. It's all completely covered over and contaminated with rhizopus, every little bit of it. And it's all asexual. There's no zygospores on any of it, right? It's just making zillions of these things. They really won't make much, because you need two different mating strings too, and there aren't two different mating strings, so it's just one string, so it's just making tons and tons of spores, but it infects everything. It's growing all over every plate you guys had. That's why you didn't see Allomyces on the practical, did you? All totally contaminated. I couldn't get any Allomyces off the plates at all. I wanted to put it in there, but it doesn't look like this. If I try to mount this, it looks like this right here. That's all there is on the plate anymore. It's killed off all the Allomyces. Yeah, so does that help kind of? I mean, honestly, I go back to the slides. I don't like go to Brooker or Campbell or something like that and start trying to find questions. Instead, I go back to the slides and try to think of what questions I could ask you based on
That'll
the
still
slides.
help us, right? I mean, reading the chapters and stuff?
Well, I think so, and I'm hoping that, like, as you go through the book, if you find things that you think conflict somehow with what I'm saying, that you'll bring it to my attention. I don't think so. I think, and I mean, more or less I'm following the book, right? I've had people say I don't use the book at all. I'm not sure exactly why, because I do tend to say Campbell and Brooker, and things like this came out of Campbell, right? And this came out of Brooker, right?
Yeah,
I mean,
I
so.
was looking through the book and I saw everything was, it's like, wow, I should read this.
Yeah,
It's just
but
everything, the lab manual and everything.
But I have had people tell me I don't use the book, and I think it's because I don't assign questions out of it and collect them or something. I realize I don't really want to do that, right? I figure, but if you want to answer the questions, that's good. I mean, I go to the back of the chapter, it couldn't hurt, right? And see if you can answer some of the questions that are in the back of the chapter. I would never use those on the exam. I'd make my own questions up, but I think that's good practice, right? And if you don't know what the answer is, come ask me. Say, I was working on number 4 here at the back of the chapter, and I don't know what the answer to this is. Can you help me figure it out? So that kind of works. So I'd go through these. There are not that many sets of slides, right? There's only like 6 or something. I'd go through those and kind of just look at the slides and just think for each one too, because this is what I kind of do. Like I'll go through here and think, okay, could I take like this picture? If I go copy, paste, and maybe even, sometimes I will do this. You'll see it when I do the animal ones. There's a lot of diagrams, like I show you like a hypothetical mollusk or something like that. And I'll just take this picture and I'll delete the words and just ask you to fill in the words. Most of you wouldn't have put that in your cheat sheet. You don't have any idea which one I'm going to take. So it's not a copying thing. You'd actually have to be able to label stuff. So here I probably expect you to be able to label R muscle and plasma membrane of the plant cell, maybe root cell. You really should be able to label all that probably. Yeah. Yeah. No. Yeah, I can show you the ones that I know I've done that before with, but they're animal ones, the ones that I can remember for sure.
Do you know why the hyphae doesn't go all the way into the plasma membrane?
Well, if it penetrated the plasma membrane, it would kill the plant cell. So it's about as intimate as you could get without actually killing the plant cell.
So I guess they just know how to back off. I guess they just know.
They know how to develop a very intimate relationship without killing their partner, right? Because if they killed their partner, that would kind of defeat the purpose. So if yeah, you can't penetrate the plasma membrane without killing the cell. This, I've used this before. You might see it on the practice exam. I've used it, I can't remember if I used it last semester or not, but I just go copy, paste. And of course, they're all putting things like Salome and stuff. There's no Salome here, obviously, but they're confusing it with other animal groups here. But it just deleted all the names, but you'll be able to put the names in. So I do this a lot with the animal ones more for some reason, but I could do it with the others. I mean, that's very often the way I'll do this. There's a lot of slides here, so obviously the plant stuff is probably a little bit easier, but I can come through here and, like here maybe. I could cross this over here, right, and then just ask you, you know, what's the difference between this and this or something. Ask you whether this is haploid or diploid. Ask you whether this is homophallic or heterophallic. There's lots of questions I could ask you there. Right? I'd have to get a little bit more creative. But again, my thinking is always, okay, what can I ask them that there's no way they could have the answer on their sheet sheet? That's really the way I think about it. That they couldn't possibly have it.